The University of Michigan Division of Gastroenterology and the Inflammatory Bowel Disease Program host the IBD Visiting Professor Lecture Series in Ann Arbor, Michigan. This series presents the latest in clinical research and basic science from IBD experts from around the world.
So thank you, and I had a great day yesterday meeting everybody, and today I'll meet a few more people. It's been quite a whirlwind, and today I did not wear comfortable shoes based on your recommendations. So I'm going to be talking about the role of immune markers in predicting disease course and therapeutic outcome. Really, the way of the future is for us to be able to move forward, and I know a lot of work is being done on imaging and looking at structural damage here, so you have that same mantra of really prognostication and thinking about natural history and thinking about the right patient for the right therapy and really attacking them early with our more effective interventions. So I'm going to take a different slant than the radiologic and fibrostenosing perspective and really talk about genetics, or touch on genetics, but really focus on the immune reactivity that individuals with IBD actually express and whether these immune reactivity markers could actually be used to think about predicting outcome in patients with IBD, particularly Crohn's disease, but there's also some emerging data in ulcerative colitis. So when we think about inflammatory bowel disease, we as clinicians are on this end of the spectrum. We see patients, and we've realized as you see more and more IBD patients that there's really a spectrum of presentations. You have some patients who present with very simple, non-penetrating, non-stricturing small bowel Crohn's disease and stay that way for a long time, and would have even stayed that way likely if we gave no medications, to be honest. There's a certain subgroup of patients who just live with mild aphthia in the ileum and cecum. A lot of you may find it when you do your colon cancer screening and you don't know really what to do with these patients. They're asymptomatic, but they have inflammatory changes under the microscope. As well as to your eye. And so what we've come to understand, there's not just one form of inflammatory bowel disease, there's many IBDs. And each of these inflammatory bowel disease phenotypes are actually represented by an underlying genetic and or immune reactivity that is specific to that phenotype that that patient is presenting to you. So a lot of us live over here. But those of us who are in the translational component of research, we're really looking for surrogate markers or other ways, subclinical, before a patient actually gets to be strictured or penetrated, where we can actually say, you're on that path, and therefore we'd like to give you early effective intervention and stop that progression in natural history for you to develop stricturing or penetrating disease. So my area of interest is really in the immune response component. There's a lot of individuals, including myself, who are also interested in the genetic susceptibility piece, but it's thought that maybe these immune reactivities are actually an expression of the body of what was going on at the level of the mucosa as a gene defect was interacting with a specific bacteria, and you were spitting out an antibody against that bacteria. That's the theory behind it. Think about it like celiac disease. You have a genetic defect. You have an underlying antigen, which is dietary as compared to an IBD, it's bacterial, and you have an antibody response to this antigen. It's kind of like that analogy in a very simple way, that you're actually expressing an antibody against a specific bacterial antigen that may be causative. Unfortunately, we're not quite as sophisticated to actually say that this particular antigen that drives this antibody response is what causes Crohn's disease. If you go back in time, historically, one of the antibodies, which is ASCA, when it was first uncovered, which is anti-Saccharomyces cerevisiae, they actually thought in Europe that Saccharomyces cerevisiae was causative because we found antibodies to a yeast in patients with Crohn's disease. So they eliminated yeast from the diet and recognized that it has nothing to do with yeast and that yeast was not the causative antigen in these particular individuals, but they realized that these must be some kind of cross-reactivity. And what I mean by cross-reactivity is ASCA is what we measure a lot. It's one of the most popular and most significant markers of immune reactivity in Crohn's patients. But we don't really think that what is the antigen or causative or driving antigen in the body is actually the yeast. We feel what's happening is that on the cell wall of this yeast, of Saccharomyces, there must be cross-reactivity with a bacterial sugar, a sugar on the cell wall of bacteria. And what this antibody is representative of some overlap or cross-reactivity with another antigen on the cell wall of a very important bacteria. And I say important when I show you how critical ASCA is in terms of predicting outcome. So the ones that are actually commercially available in the US are most of them actually, but the ones we are familiar with are the top 5 here: Pienka, ASCA, OMSI, and CBER. These are actually available through Prometheus labs. ASCA and Pienka is often available through other labs. It's, the Pienka is different based on different labs, but ASCA is essentially the same wherever you get it. These antibodies, which are called anti-glycan antibodies, are by a company called Glycamid, which is based out of Israel. They're actually not a commercial lab with a sales force, but they go through LabCorp. So I don't know if you have access. Peter, do you have access to Glycamid? Or?
It's painful, but you can do it.
Okay. So Glycamid, because they're not a company that markets, they just actually go through LabCorp. So some labs, if they have contracts with LabCorp and wouldn't send somewhere else, you would get the anti-glycan antibodies as a result. So these are anti-glycans, which is very similar to ASCA, which means an antibody against a sugar in the cell wall of bacteria and or yeast in this particular situation. This is an antibody against a flagellin. This is an antibody against a specific outer membrane porin known to be localized on E. coli. Pienka was actually thought to be an autoantibody, but now actually people think that it's truly an antibody against a bacterial component. So there's increasing interest in understanding what these antibodies represent, because maybe there's actually a causative antigen. As we know, it's not very simple in IBD where you have one antigen, one gene, one phenotype, or else we wouldn't be here, right? So the reality is it's much more complex than that. So that's why the knowledge of multiple immune reactivities is actually interesting, because it means and it talks to the multifactorial nature of IBD. And to think that each of these probably represents some kind of gene-bacterial interaction is very interesting, because it lends itself to the hypothesis that we believe IBD results from a genetic susceptibility and an inappropriate handling of our bacteria. So it's kind of interesting to think that we may have actually markers of this abnormal handling of bacteria in specific gene defects. So if we're going to phenotype patients or classify patients much deeper than just clinically, meaning you have small bowel, you have stricturing, you have penetrating, you have a family history, you're diagnosed below the age of 16, these are all classification systems we use to phenotype. We could think about actually phenotyping patients at a subclinical level, meaning at the immunotype, rather than the clinical phenotype. We can think about classifying patients at the immune level. And so back in 2002, Carol Lander's group, they actually started to think about how do Crohn's patients kind of end up in terms of how many antibodies each of them have, and what's the combination of these markers that an individual can have. And what you can see here is that there's about a quarter of patients who are positive for all 3 of these bacterial-driven antigens, or antibodies against bacterial-driven antigens. The question will be, which I'll share with you in a few moments, is what does that mean to be positive for all of these markers? Does that impact the way I view my patient, and does that impact the progression of disease? Meaning, is it a bad thing or is it a good thing? And I'll share with you the data on that. And then you can see there are patients who have overlap of 2 of these, and then there are individual patients who have the remaining difference of the antibodies that are individually positive. Then in 2005, CBER came along. CBER was interesting because it was actually found in an animal model at Chuck Elson's lab in Alabama. And then he said, hey, take this antigen and actually test it in your humans and see whether or not your human Crohn's patients actually have antibodies against this antigen. And 50% of Crohn's patients were positive for this antibody. But it was actually identified in an animal model that got inflammatory bowel disease. So it thought, wow, this could be causative. There could be something about this flagellin. And it's a Clostridia flagellin. It's not C. diff because everyone got excited, but it is not. It's actually, the family of Clostridia is huge, and this is just one of the family of, or species of Clostridia.